Severe acute respiratory syndrome coronavirus 2, the cause of the coronavirus disease 2019 pandemic, is a single-stranded positive-stranded RNA associated with the respiratory, gastrointestinal, and central nervous systems virus. Infection in humans. Infection is mediated through the interaction of membrane-bound human angiotensin-converting enzyme 2 with the viral spike protein, and is further aided by host proteases TMP2, TMP4, and CatB. ACE2 has been reported to be expressed at the brush border of enterocytes in the midgut, which is also the site of SARS-CoV-2 related inflammation. Post-acute COVID-19 syndrome, characterized by symptoms that persist or prolong beyond 4 weeks after infection, was initially ignored by many. However, it is now considered a multi-organ disease that poses a growing public health challenge. Symptoms associated with post-acute COVID-19 include severe fatigue, cognitive impairment, or pain. Severe acute COVID-19 and poor baseline health often lead to post-acute COVID-19, although mild infections can sometimes lead to post-acute COVID-19. Pathophysiological mechanisms after acute COVID-19 are not as well defined as in acute COVID-19. Furthermore, post-acute COVID-19 has been reported to be associated with hyperactivated T and B cells, persistently activated innate immune cells, and increased expression of pro-inflammatory cytokines. However, the cause of this immune disorder is unclear. A new study in the journal Gastroenterology uses upper and lower gastrointestinal endoscopy to assess whether persistence of gut antigens contributes to acute COVID-19 in patients with or without acute COVID-19 symptoms. Study: Post-acute COVID-19 is characterized by the persistence of viral gut antigens in inflammatory bowel disease. Image Creative Nico. About research. The study included 46 inflammatory bowel disease patients with PCR-confirmed SARS-CoV-2 infection. Intestinal tissue was obtained by upper and lower endoscopy 94 to 257 days after infection. In addition, all participants had to undergo laboratory testing and quantification of fecal calprotectin at the time of endoscopy. Biopsy specimens from the terminal ileum, duodenum, and colon were used to determine the persistence of SARS-CoV-2 RNA in the gastrointestinal tract, followed by immunofluorescence. Thereafter, Vero cells overexpressing ACE2 and TMP2 were infected with the virus. Quantification of SARS-CoV-2